So this algorithm trace that we developed takes the single cell repertoire, both the TCR repertoire and the RNA-seq repertoire from the starting material, which is the tumor harvest or in the in situ TILs, analyze the gene expression patterns and builds a machine learning model to predict at a single clone level whether that T cell is more likely to be tumor reactive versus not. We have leveraged a lot of the data that other groups have published. So we are very grateful for those studies, but also we did other things such as annotating our training data sets with obviously non-tumor reactive TCR clones such as viral reactive and so on. Plus we supplemented our own internal training data to build up a fairly large training data set to build this machine learning model. I'm Dipen Sangurdekar, Head of Data Science at KSQ Therapeutics, and you're listening to the Repertoire Room.
Hi, Dipen. That's a very good intro. Thank you for doing that. And thank you for joining me on this podcast on T-cell repertoires. In this show, I actually want to have insightful conversations with people who share a passion and go deep into the T-cell sequencing technologies and analysis. I know you're also into the single cell, so we can briefly also touch upon that. It's not only have to be about TCRs, but I'm really excited to have you here, especially because I saw a recent paper published by you and your colleagues, and we can talk a bit maybe about that later on, the trace paper. But before we dive into who Dipen is, can you maybe shortly introduce your company and KSK Therapeutics?
Absolutely. And first of all, thank you for having me on this podcast. It's always a pleasure to talk science with colleagues in the field. KSQ Therapeutics is a clinical stage company. The company has been around for 10 years. We are based in the greater Boston area. KSQ actually stands for Kindle Square, which is where the company was founded. The scientific founders of the company from MIT, the Broad, and the Whitehead Institute, and UCSF, they had a vision, and the vision was to leverage high throughput CRISPR screens very comprehensively, so genome-wide across a variety of tissue and cellular context. And the goal of that was to unbiasedly find the right target in the right context. And the team, the founding team, the KSQ initial team, actually pioneered the way of industrializing CRISPR screens. Before anyone else did large-scale screens in a variety of contexts, from cancer cell lines to immune cells. And what this led to was a number of novel targets, targets that no one else had described before, and actually which have been subsequently validated by other publications in academia and industry. So, of course, finding targets is one thing. We have to still validate the targets, convert them into therapeutic hypothesis, and the company has successfully done that for not just a single modality, but multiple modalities, from small molecules to now our current focus in cell therapies. So starting with small molecules, the first program we took to the clinic was a first-in-class target, USP1, which is discovered in a synthetic lethality context in unbiased CRISPR screens across hundreds of cancer cell lines. And that small molecule program was taken to phase 1. Our current focus is actually using CRISPR technologies, not just a discovery platform, but actually a therapeutic modality as well. So what we do is we have identified best-in-class targets in T cells, in CD8 T cells, and we are now using that as an ex vivo gene-engineered cell therapy hypothesis. And what that means is we take patients' own T cells, they're harnessing their immune system, so the T cells that are exposed to their tumors and have been trained to fight the tumors. And every cancer patient have T cells, but with varying levels of success fighting the tumors. But what we do is we extract those T cells from the tumors, from the solid tumors. We engineer them ex vivo and knock out specifically one or two genes, which we think will, or which we believe will impart with a set of desirable attributes to those T cells, from they're enhancing their proliferative capacity, reducing their dependence on cytokines, and their ability to persist in fairly hostile tumor environments. And we infuse those cells back into the patients and those T cells are now supercharged. They're polyclonal, and now they're also trained to identify the tumors. So this class of therapies is called TIL therapies, tumor-infiltrating lymphocyte therapies, and they are sort of a slight variant on CAR T therapies, which are far more sort of well known. But fun fact, right? The first autologous cell therapy that was approved for solid tumors was a TIL therapy, and that's because they tackle directly the solid tumor heterogeneity challenge. So yeah, so that's what the company is, and we've been sort of in the genomic discovery and engineering space for a long time, and we're one of the leaders in that space.
This was surprising during my research for the company because I actually got to know KSQ as a TIL company, but then digging deeper, it's basically a molecular engineering engine, CRISPR technology to find targets, and then all of these therapeutic modalities. It really surprised me because I first got to know you just because of the TIL-focused posters, and it's only then digging deeper, like, oh, it must have been an interesting challenge as well over these different modalities, but it's really cool to have this powerful scientific engine actually to do the therapeutic target discoveries. Yeah. But yeah, I guess also challenging going across different modalities. Although the focus seems to be TIL for now, and I think it's you're getting pretty well along there. I'd love to go back to that and maybe what's in the pipeline later on. But I'm also always interested in my guest's personal background because people just don't end up in biotech companies by accident, I think. Most of them have an interesting career also before that. So I'm very curious to know what's your origin story and how did you actually, what has your career been prior to your current role at KSQ?
Yeah, no. So it's been quite a journey. I was in my undergraduate and my early graduate career, I was trained as an engineer, actually a chemical engineer. And chemical engineers tend to be a diverse lot, and are cross-trained in multiple ways of thinking and tackling a problem, from the physical sciences to the life sciences. And yeah, my curiosity took me to pick up a pipette and be trained as a scientist. I actually cross-trained as a scientist and a computational biologist through my PhD thesis. I actually worked on uh, understanding the underpinnings of antibiotic resistance in bacteria and control of metabolism using transcriptional and genetic control. And as I was doing the experiments due on my engineering background, I started publishing or developing computational methods, machine learning methods to analyze the data. I parlayed that into postdoc, which was again cross sort of between developing statistical methods and also setting up new experimental methodologies for high throughput genetic genotype phenotype association analysis. Eventually, I decided that my computational skills were far superior than my experimental and pipetting skills. So I left that to the folks that had the magic of doing the right experiments, and thinking through the experiments and doing them very efficiently. And I decided to focus on computational biology and bioinformatics, and have been in the industry purely in that realm for about 15 years now. Across a number of different companies, both large and small. So my work broadly has spanned two therapeutic areas: oncology and neuroscience. And um, while I've worked across the R&D pipeline, I've sort of more focused on that messy preclinical to clinical translation interface, right, where we take our preclinical mechanism of action hypothesis to the clinic and then see what happens. And it's in that stage where you discover something new every day. And of course, also learning from the clinic, taking clinical insights and taking them back to generate new scientific hypotheses and so on. So that's been me. I've been with KSQ now for about 4.5 years.
Yeah. Oh, we share an initial PhD background then. I actually also started my PhD in microbiome research, so microbial research and doing whole genome sequencing and analysis of microbes. And this is also where I learned that a pipette is interesting, but not my best asset. And it's interesting that you only made the switch at a postdoc level. You still went through the whole PhD then doing a lot of wet lab screenings, I guess then.
And postdoc as well. So my postdoc was also about 50% dry lab, 50% wet lab. So it was a fantastic experience learning from others in the field. And again, kudos to all the scientists who actually do those beautiful experiments and generate the great data so that, we computational biologists have something to analyze.
Yeah. There's also a strong asset in being part in the wet lab and part in the dry lab, because then you also get to know how these kind of experiments are being generated, and maybe the pitfalls in the data analysis as well.
Oh, that's a great point. And this is such an important thing for, when we talk about AI and machine learning and AI-ready data sets, right? So what's often lost, I mean, not really lost among the practitioners who do this day in, day out, but what's less obvious is all the things that go into making a data analysis worthy, right? Forget AI worthy. And unless you have a deep understanding the technology, the assays that generate the data, you don't understand the confounding, the batch fix, the quality, the negative space in the data, right? And if you're close to the bench, you develop an immediate intuition for it. But successful computational biologists that I work with either have been on the bench before or have worked very closely with folks on the bench. And that's where you fully understand that it's not just a matrix of numbers, it's like a living, there is a story behind it, right? How that data got there. So absolutely agree.
Especially in biology, right? Indeed, there's so much that can happen, and that happens. I fully agree with that statement, actually. Okay, yeah. Back to KSQ. The portfolio you mentioned already, there's the ETILs up in there. Can you tell me a bit more what you currently have in the pipeline? What kind of assets?
Yeah. So as I mentioned earlier, using large-scale CRISPR screens, actually targeting all 20,000 genes that we could design CRISPR guides for. The team, the research team at KSQ and the discovery team did extensive CRISPR screens, not just in vitro in a plate, but in mouse models and on mouse human models, and identified genes that not just increased T-cell infiltration into the tumors and the persistence, but then during validation process demonstrated that those targets or knocking out those targets actually enhance the cell's ability to kill tumors. So they did this not just in a univariate way with one gene at a time, but also in a combinatorial way to find best pairs of genes that you can target to maximize the cytotoxic potential, but also the persistence and health attributes of those cells. All of that work is now either published or in the process of being publication. Actually, we just put the the combinatorial screen work as a preprint on bioRxiv, so please look it up if you're interested in learning more. The single edit or the single gene that we found first, we actually took that into the clinic as a single edited TIL therapy program or ETIL therapy program that was called KST01X, 001EX. And that gene is SOCS1, which is a suppressor of cytokine signaling. It's a very well studied and understood gene. It's a negative inhibitor of JAK-STAT signaling pathway. And what that does is knocking out that gene inhibits the feedback loops that cells have when they see a lot of cytokine-mediated signaling. So they are basically taking the brakes off the cell that allows the cells to be hyper-responsive to cytokines and produce more effector cytokines as well. So KSQ01EX is in the clinic. It's been in the clinic for a couple of years now. The second program that we have in the clinic, KSQ04EX on the ETIL side, has two genes knocked out. One is SOCS1, and then its best combination partner, REGNASE-1. So SOCS and REGNASE are the two genes that we're knocking out for the KSQ-4EX program. And again, these genes are not just well understood. REGNASE, actually, when we discovered it, or when the team discovered it many years ago, was less well known. But since then, a number of people and academic groups have published REGNASE-1 as a top candidate. And REGNASE-1 is an RNase, and its targets include genes that are involved in the cytotoxic functionality of the cells. So by knocking out REGNASE-1, we are allowing its targets to its gene targets to be upregulated and allow those cells to persist, proliferate, and have a less differentiated effector-like phenotype rather than an exhausted phenotype. So KSQ-4EX has been in the clinic for over a year now, and we also just read out some data from the single edited O1EX program at ACR last month.
Yeah, these are from a molecular biology standpoint of view extremely interesting. Like you do the single edits, but also the combination of two of them, bringing them into clinic, and how have these responses been? Like the single one is already was already in clinic. Has this been a good pathway for KSK?
We reported some clinical data at ACR from the KSQ-01EX study. Again, early phase 1 studies in oncology tend to be efficacy studies. We want to make sure that cell therapies, we are not seeing any adverse events. It's also, you want to start at lower doses. There are other nuances with TIL therapies. Like, for example, we have TIL therapies as pioneered by Steven Rosenberg's group at NCI for many decades now, have adjuvant IL-2 treatment, which has its own toxicity profile. So we want to make sure that whenever we do gene engineering on these therapies, we want to start small. We want to make sure that we're developing a safe product. So we sort of gradually ramp up both the dosing and play around with certain parameters. So early data on the single edit study shows that the drug is safe, and there are hints of efficacy that gives us a lot of reason to believe, not just in that program, but also the dual-edited program.
Yeah, that's exciting. Like having two different kind of slightly different therapies going out there. The first signals of efficacy, of course, I know phase 1s are not designed to do that, but it's exciting to see that you can get along, and very, very interested to see where this will bring you in a couple of years as well. Now, Dipen, one of the reasons why I wanted you on this podcast is because I actually saw a paper of you, the TRACE paper, where you actually introduced a machine learning model to the world. I'd like to talk a bit more about that one. I know it's not about T-cell sequencing per se, but more about maybe it does include T-cells, right? So we can talk about that. But can you tell us a bit more what TRACE exactly does and how it has been applied within KSQ?
Yeah, so the problem we're trying to solve comes from one of the best attributes of TIL therapies, right? So unlike CAR-T therapies, where you're inserting a chimeric receptor to go after a single antigen target on tumors, right? We know that solid tumors have the ability to express multiple antigens. A lot of the surface expressed markers are not necessarily lineage markers, so cancer cells can downregulate those and still be very proliferative. Also, there are multiple of those markers that cancer cells can express, and therefore TILs are very powerful, or have that advantage over CARs, is that the TILs are polyclonal. That means the TCR and the native TCR sequences on those T cells have been selected by the patient's own immune system to identify those new antigens or tumor-associated antigens and kill the cells. Now, of course, a lot of the times that the natural immune system falls short, not just because of the complexity of the tumor microenvironment, the immunosuppressive nature, but also the inability of the T cells to adequately overcome those challenges and affect tumor killing. So the cancer cells basically outgrow or outproliferate the T cells. But the inherent design of the anti-tumor activity is there, right, in those T cells, or those T cells have been trained or selected for that. So what we do is we take those T cells out and we grow them ex vivo and put them back, so that now instead of having a few thousand or a couple of million cells, we have billions of cells that contain those tumor-reactive T cells. Now that brings me to the crux of the problem, right? Ours is an unselected TIL therapy. That means we take all the T cells we can find from the patient's own tumors and grow them out. Now, not all TCRs or T cells will have TCRs that are tumor reactive. While what we have seen and what's been reported in the literature is anywhere from low single percent fraction to high double-digit fraction of those T cells can be potentially tumor reactive. So it's of interest for us to understand our drug product. The drug product is the TIL therapy product that we manufacture and infuse into the patients. We call it the drug product. For us to understand how potent or how effective that drug product is going to be, we need to understand which of those TCRs, of thousands or tens of thousands of TCRs that our T cells in the drug product contain, which of those are tumor reactive. And that's a very hard problem to do, to solve just by looking at TCR sequence alone. Fortunately, when we look at the T cells that are in the tumor context before we harvest them, because those tumor-reactive TCRs or tumor-reactive T cells are constantly exposed to the tumor-associated antigens or neoantigens, those cells occupy a certain transcriptional state. So regardless of what the TCR sequences are, right, because they are constantly fighting the tumors, they go down this certain differentiation state, right, exhaustion, terminal exhaustion, and so on. And it has been reported by other groups for several years that those transcriptional states are easier to identify using technologies such as single-cell RNA sequencing. Right? So there are certain canonical gene expression markers that are observed in those cells that are known to be tumor reactive. One of them is CXCL13, which basically every study that has looked into this has found it. Right? Other surface markers include CD39, CD103, and so on. So we want to take advantage of that fact that there are certain gene expression markers that define the transcriptional niche that can potentially separate out tumor-reactive TILs from non-tumor reactive TILs or viral reactive TILs or bystander TILs, and develop a classifier. Now, to be perfectly clear, we are not the first group to think of this idea, and there have been several groundbreaking publications in this space from NCI and from other groups that have built those methods, and we are basically standing on the shoulders of those giants to advance the field a little bit further. So this algorithm trace that we developed takes the single cell repertoire, both the TCR repertoire and the RNA-seq repertoire from the starting material, which is the tumor harvest or in the in situ TILs, analyze the gene expression patterns and build a machine learning model to predict at a single clone level whether that T cell is more likely to be tumor reactive versus not. We have leveraged a lot of the data that other groups have published. So we are very grateful for those studies. But also we did other things such as annotating our training data sets with obviously non-tumor reactive TCR clones such as viral reactive and so on. Plus we supplemented our own internal training data to build up a fairly large training data set to build this machine learning model. So that's the origin story of Trace. We have, in recognition of how much we were able to do this because of the community work that has happened, we actually made the model weights, the code completely open source, so it's free for anyone to use and adapt and develop further and retrain on their own data as well. We really see this as as a joint effort, right? And we are all fighting, fighting the same battle, right, against cancer. So we wanted to make this method as easy to use, but also as comprehensive as possible.
Thank you for that summary, and also thank you for pointing out indeed the work that others have been doing in that field. And it now makes sense to me why you would publish this, because I think, oh, this could be a very cool asset for the company as well to be using. But it's also the gratefulness for the other people and what they've been doing that actually was the motivation to also put it out there. Yes. Okay. Now, we talked about trace, single cell, and we talked about TCRs as well. Maybe just to make the full link with the topic of this podcast, do you sometimes also just look at TCR repertoires, for example, in clinical samples or like preclinical, etc.? That's also part of the research tools at KSQ?
Absolutely, absolutely. So we extensively use bulk PCR sequencing for multiple reasons. One is, as we are developing our products, we want to make sure that we don't lose the best attribute of our TILs, which is the polyclonality. So we don't want to accidentally invent a manufacturing process which makes our very polyclonal, heterogeneous TIL repertoire into an oligoclonal repertoire. So we don't want just to accidentally expand a single or a limited set of clones. So measuring the diversity of our repertoire at the end of the manufacturing process is a key attribute that we look for every single time. And whenever we do process changes on any experiments, we want to make sure that we maintain that richness and polyclonality of our TIL products. On the clinical side, we use bulk PCR sequencing methods. Post-infusion of our drug product into the patients, we track the repertoire of T cells in the patient's blood to track the clonal dynamics of their T cells, post treatment. So the idea behind that is we want to see the pharmacokinetics of our drug product in the patient's blood. Now, it's a harder problem because obviously when we infuse the drug product, these patients are lymphodepleted. So initially they still don't start with any of the T cells of their own. So for the first few days, all the T cells in their body or in the periphery are from the drug product. But after a week or 10 days, right, their indigenous repertoire starts coming back. So then you get a mix of your whatever is left of your drug product in circulation and their indigenous repertoire. So tracking those clonal dynamics, right, of how our repertoire, the DP drug product repertoire does compared to the endogenous repertoire. Are there certain clones that are expanding? Does it correlate with clinical efficacy and the quality and the durability of response? And since we can actually use trace to identify tumor-reactive TCRs in our drug product, starting from the tumor harvest itself, we can actually track how those tumor-reactive clones are or potential tumor-reactive clones, right, are doing in terms of dynamics post-infusion into the patients. And actually, we also oftentimes take a biopsy of the tumor a few weeks after treatment. Hopefully, all our patients are cured and we don't have any biopsies or any material to biopsy. But sometimes, that's not always the case. And we do look at biopsies, and we'll do look at T cell infiltration in those biopsies after treatment, and we can actually track both using a TCR repertoire. But also, I didn't talk about the fact that since we're knocking out genes, we can actually track using NGS. We can actually track the gene edited cells in our tumor as well. So this leads to a very nice multimodal data cube, and we spent a lot of time thinking and analyzing that data and sort of drawing the connections between mechanistic hypothesis to what's actually being observed in the clinic.
I have to admit now that now I'm starting to understand what a VP and head of data science at KSQ has to do. There's a lot of data coming in from preclinical to like NGS data from, like you said, the gene editing to really like PBMC samples from clinical samples and their TCR sequencing on. It's probably just only a small drop of maybe the kind of data that you touch, but it's exciting that you're working on all of these levels, the preclinical one, or like the drug product itself, and then having that data to then compare what's going on specifically in the patient and see how this repertoire evolves over time and how your drug product actually will maintain over time. It looks like there are interesting data sets going around at KSQ from my perspective.
No, and that's immunology is so complex, and you're also very close to the field, so you appreciate, right? We can only get you can get multimodal representation of the T cell, but it's never complete, right? Each modality
I
doesn't
know.
give you a
Yeah.
complete representation. So it's almost like you're piecing together what the cell phenotype actually is based on these orthogonal pieces of evidence, and you're drawing inference either quantitatively or subjectively based on those different pieces of data. So it's very fascinating. And actually, one area of research that I'm very excited about is multimodal foundation models, right? Which are actually already making an impact in the clinical space with patient records and their H&E stains and the genetics and so on, right? Building those multimodal foundation models are more predictive, more performed than single modalities alone. And I'm looking forward to these multimodal immunology foundation models coming up. I know a few companies or academic institutions are working on that, and those will be very powerful as well for us to get a better picture of what our cells actually are in their quote unquote native state versus in an engineered state. So.
I agree. That's a very exciting avenue for the future. But I also know from my experience as a postdoc looking at multimodal views at things. In my case, it was multimodal views at microbiomes. It's also not easy. Like indeed, a single modality already comes with its challenges. Combining everything together and maybe how the data is being generated in the lab always has its challenges. But I'm pretty sure indeed that the way that machine learning and AI is evolving, that that it will become easier in the future, although I don't think it's something already for tomorrow. But I guess we still have a couple of steps to take before we really get there. Okay, Dipen, this has been extremely insightful for me. What is actually next on the agenda for KSQ in the coming years? What excites you?
No, so we like to think that we are at our heart still a genome engineering company, right? Not just engineering for creating new therapeutics, but also understanding biology, right? And I feel very proud of what the company has done in terms of translating those novel biological insights in actual drugs that work in patients. And we will continue on that mission, right? Both on our ETIL space, but also in other modalities, edited cell therapy modalities, not just limited to not to TILs. So yeah, no. In the short term, we are keenly watching how our clinical studies are progressing, and as we go through the traditional clinical development stages, right? Dose escalation, making sure that drugs are safe, check efficacious, check finding the right dose, right? So that will be something that we were, at least my team and we are working closely on that data and sort of analyzing the data and making sure that we are making the right decisions based on that data. So
Yes, I'll keep a good eye on what's coming out in the next years. But this has been very rewarding for me. It's nice to talk with people knee deep in NGS technologies and using that really to bring things into the clinic and analyzing clinical data as well. So thank you for your time. Thank you for being here. One last maybe closing question. Where can people find out more about you and KSK if they should want to connect or have follow-up questions?
yeah, as with everyone, I have a profile on LinkedIn, easy to find, and KSQ, the website is ksqtx.com, and of course, feel free to follow the company's LinkedIn and find more information. We do publish all of our posters and recent scientific work on our company's website and oftentimes in LinkedIn posts as well. So all that information is out there.
Great. Again, thank you for being here. Thank you for being my sixth guest in the Repertoire Room, and we'll definitely connect again in the future.
Thanks, Shander. Thank you for having me. It's been a pleasure.